Okay, well, thank you very much. First, to those attending in person to this seminar, and also people who is connecting remotely. Well, it's my pleasure to introduce Dr. Quynh, with whom well, I have been collaborating in the last years in the framework of different initiatives. Dr. Quynh is a researcher at the Institute of Agriculture and Environment in Vietnam. And well, we have been involved in, together with other people from his team and also people from COAPA at CEIGRAM, as Guillermo Guardia, who is there. And Guillermo has kindly offered himself to chair the session on site, so he will be the chair of the seminar for questions later on. We have been collaborating with Quynh and colleagues in a research coordinated project of the International Atomic Agency and FAO, focusing on climate smart agriculture, looking for strategies to reduce greenhouse gases from croplands, as well as increasing the carbon sequestration in these agroecosystems. And today, well, Dr. Quynh is gonna present us some of the, well, the main outcomes and features of our research carried out in our rice field in Vietnam, in which he has been assessing or testing different fertilization and other management practices to decrease reactive nitrogen fluxes to the atmosphere and to increase the nitrogen use efficiency in these systems. So, yeah, now, well, of course, all this has been possible with the supervision of our colleague, Mohamed Saman, who is actually, well, is recovering from a health issue. So I think Quynh will be agreeing with me that this seminar could be on, yeah, acknowledging, dedicated to Saman and acknowledging his work on these initiatives at the International Atomic Energy Agency. So the floor is yours, Quynh. Thank you.
Yeah. Thank you, Professor Alberto. And good morning, everyone. My name is Quynh. I come from Vietnam. I work for Institute of Agriculture and Environment. I under the Vietnam Academy of Agriculture Science, under the Ministry of Agriculture Environment in Vietnam. So on behalf of my team, I would like to present you some results from my field experiment in Vietnam. So firstly, I would like to provide you the context of rice cultivation in Vietnam. You know, my country, the main crop is rice. So the rice cultivation area, we have 3.2 million hectares. 3.2 million hectares. So in some area, the farmer, for example, in the north of Vietnam, the farmer have 2 rice crop per year. But in the south of Vietnam, the farmers sometimes they have 3 rice crop per year. So meaning that the planted rice area reach to 7.2 million hectares of rice. So we are also, sometimes we are the first export rice country. Sometimes Thailand is the first, and we are sometimes is the first position, and sometimes is the second rice export country. Every year, you know, the total rice production every year we produce around 43 million tons of rice, and we export around 6 to 9 million tons of rice every year. If you go to Vietnam 30 years ago, so that time we are very poor. Even we lack of rice, but now very lucky we can export 6 to 9 million tons of rice every year because the government invests a lot for new technology, fertilization, and so on. So therefore, the rice is increasing. But however, rice also emit a lot of greenhouse gas emission. Let me show you in every year, Vietnam, the total greenhouse gas emission from whole country, from whole sector, around 360 million ton of CO2 equivalent, in which 33% come from agriculture sector. 33% is around 88 million ton of CO2 equivalent. In agriculture sector, 50% GHG emission come from rice, paddy rice cultivation. Is around 44 million ton of CO2 equivalent emit from paddy rice. So therefore, we give a lot of efforts to reduce greenhouse gas emission from paddy rice field, especially for methane emission, because paddy rice anaerobic condition, so emit a lot of methane emission from paddy rice field. You see, 50% come from agriculture, come from rice. So we give a lot of effort to reduce greenhouse gas emission from paddy rice field. The most important mitigation is water management. So we in traditional rice cultivation, the farmer only think paddy rice only needs the water, so they keep the field flooding for the whole dry season. So therefore, methane emission is very high. But so therefore, we try to convince the farmer apply AWD or mid-season drainage. Mid-season drainage meaning the farmer have one drain water out of the field only one time per whole dry season. But AWD is an alternative wetting and drying, meaning more than 2 drain during the dry season. It's a different mid-season drainage and AWD, alternative wetting drying, because dry is not always need water. So therefore, for AWD, we just keep water in the only 2 steps. The first step is the first 20 days after sowing is important. Even in that time, rice plant is very small. Rice doesn't need too much water, but we need to keep water in the paddy rice field to reduce the risk from the weed and rust. Is a problem. Weed and rust. If you don't keep the water in the field, the rust will attack the young seedling, and also the weed go very fast. So therefore, in the first 20 days, we convince the farmer keep the rice flooding. Is an important thing. And the second step, we need to keep the paddy rice flooding. Is 10 days before and 10 days after full flowering. Full flowering, the rice plant require a lot of water. So in that time, we have to keep the water in the field. We cannot apply AWD. So the rest of rice growing season, we can apply AWD. What is the AWD? The farmer irrigate water in the field, keep 7 centimeter in the 5 to 7 centimeter flooding, and then the farmer will wait water nature evaporation or rice plant use water. Yeah, and then when the water go down, minus 10, minus 15 centimeter, and then the farmer will re-irrigate. Yeah, something like that. Yeah, it's a AWD. If you apply mid-season drainage, you can reduce around 15 or 20% of methane emission compare with the continuous flooding. Let's say traditional practice. If you apply AWD, meaning you have more than 2 drain per dry season, you can reduce from 30% to 50% of methane emission. This is a lot. If you can emit the total greenhouse gas emission from agriculture sector in Vietnam is 44 million tons of CO2 equivalent. So if you reduce 30%, meaning you can reduce 12 million tons of CO2 equivalent. You can generate 12 million carbon credit. Yeah. This is an important mitigation option. Second one is rice straw management. Rice straw management also very important because is a carbon input in the soil. Yeah. Now in Vietnam, especially in the south of Vietnam, we have 3 rice crops per year. It's meaning you have only 2 weeks between the rice season. It's very, very short. So how rice straw can decompose well in just 2 weeks? Yeah, is otherwise make the toxic for the next rice season for the Duong seedling. Is an important thing. And second thing is if you don't enough the time to decompose the rice straw carbon, and then the next rice season carbon will emit a lot of methane in the flooding condition. So therefore, in this we have two strategy to reduce methane emission. The first thing that we try to convince the farmer should incorporate rice straw immediately after harvesting. Yeah. Second things, we need to compost rice straw, or maybe you can produce biochar from rice straw. Yeah. And then you can apply composted rice straw, or you can apply biochar derived from rice straw. Yeah. And then especially for biochar, if you apply biochar in our research, significant biochar can reduce methane emission. And second thing, the biochar can sequester the carbon in the soil. And then you not only claim carbon credit from reduction, you can also claim carbon credit from carbon sequestration. So the sequestration carbon biochar credit have 10 times expensive than CO2 carbon credit from reduction. Yeah, but of course, if you produce biochar, you also need to invest more money than composted manure. And the third thing is you can use a short grow rice variety, or you can use the inbred rice variety, because you know the 90% of methane emission emit to the atmosphere through the parenchyma in rice plant. So meaning the more the biomass, the more methane emission. Yeah. So therefore, if you use inbred rice variety, have lower biomass than hybrid rice variety, and then you can reduce methane emission. Or you can use a short grow rice variety. Of course, you can also reduce methane emission. And the last one, nitrogen fertilizer management. Of course, it's a peri-dry system N2O emission is low if you compare with methane, but you still need to convince the farmer to apply optimize nitrogen management to not only reduce ammonia emission, N2O emission, you can also increase nitrogen use efficiency, and then you can input the economic for the rice farmer. So therefore, my finished experiment focus in the last one, because in the first three mitigation option I mentioned here, a lot of people research. I also carry out a lot of research on this three, and then now I present you some result from the last one. Here it is my field experiment. This experiment funded by IIEA. We have the same group with Professor Ambeto and Professor Gumel. Sorry, Gumel. sorry, my pronunciation. Yes, for field experiment we conducted in Vietnam 2 years ago, and the field experiment here includes 6 shipments, and the all 6 shipments will receive the same amount of phosphorus and potassium, and also the same amount of nitrogen fertilizer, but the difference is different inhibitor. Here the first shipment is no nitrogen fertilizer. The second one is urea, the normal urea is you can say is a farmer practice, the control shipment. And the third one is urea coated NBTVT, 40% of NBTVT and 5% of DCD. NBTVT is urea hydrolysis inhibitor, and DCD is nitrification inhibitor. And the number 4, the urea coated 30% of DCD and 5% of NBTVT. And number 5, urea coated polymer. And the last one, number 6, is the urea plus biochar. And then we will compare how different inhibitor, nitrogen inhibitor, can reduce ammonia, reduce N2O emission, and at the same time increase nitrogen use efficiency and yield. Is our experiment layout. We have 3 blocks. Each block we have 6 shipments. It's a random complete block design. Yeah. So we use we call the Coca-Cola methods to developed by the Professor Segundo from Brazil. He is the same our group with IIEA, and we use sulfuric acid to capture ammonia, and then we bring to the lab for analysis ammonia. This is a protocol for analysis of ammonia. So I don't want to go to detail.
Sorry, Quynh. I just placed in the chat the research paper behind this method. It's semi-open chambers instead of the name you used.
Okay. This is N2O emission. We use a static chamber. This static chamber includes 2 parts. The base chamber we put permanent in the peridifin, and the top chamber we only bring to mount is the base chamber on the cylinder. And we take the N2O emission every 10 days, something like that. For ammonia sampling, we take more intensive than N2O emission. For example, we take the day 1, 2, 4, 7, 11, and 15 day after fertilization. So we have three fertilization dressing. The first is the basal dressing. The second is 2 weeks after sowing. The third one is 42 days after sowing. So for each fertilization event, we take the ammonia sample day 1, 2, 4, 5, 11, and 15. And of course, we monitor the right use. And you can see the micro plot is 1 square meter. We apply 15 N. 15 N in this one. And right. We use 2 methods to calculate nitrogen U emission. The first one, different method. We depend on the nitrogen amount in the fertilized plant minus for nitrogen amount in the non-nitrogen fertilized. The first treatment. And the second method, we apply the isotope method to calculate nitrogen use efficiency. This is the different step how we prepare 15N to apply. So I would like to jump to the result, the main result. You can see the treatment tree. So the high gas yield is significant higher in the control, 57 compared with 46 quintas per hectare. And treatment 5, 56.5 ton per quintal per hectare, also significant higher than the control. And this is a summer season, and this is spring dry season. Also, you can see the number 3 urea coated NPTVT shows the high gas yield. Yeah, compared with the practice, farmer practice. And this figure shows you the ammonia emission. Ammonia emission here. This is ammonia emission in the summer dry season. You can see the farmer practice urea in the red line, the very high emission in after the 1 day fertilization. Yeah, in urea. And treatment 3, this is the green one, and also polymer. Urea coated polymer. So the orange line lower than farmer practice. And in the spring dry season, you can see the same urea show the high gas ammonia emission. And the summer dry season is hotter than spring dry season. So therefore, you can see the spring dry season, the ammonia emission in the first 2 weeks is low. If you compare with the summer dry season, the average temperature is around 33, 35 degrees Celsius. So the ammonia emission is very high after 1 or 2 days fertilization. But in the spring dry season, at the beginning of spring, the temperature is very low. So therefore, ammonia's emission is low, also low. The 10 degrees Celsius difference between the summer dry and spring dry season. is the cumulated ammonia emission. You can see the urea-coated polymer significantly reduce 37% ammonia compared with the farmer practice. Achievement 3 urea-coated NPTVT reduce 19% compared with the urea, normal urea. And also in the spring dry season, urea-coated NPTVT can reduce 22%. The rest reduce around 8%. The N2O emission in summer season. You can see achievement 3 urea-coated NPTVT can reduce, but it's not significant different with the control 2. But for urea blood biochar, is a significant reduce N2O emission compared with the farmer practice. And in spring dry season is achievement 3 significant reduce N2O. And also biochar significant reduce N2O compared with the farmer practice. And this also use the nitrogen U efficiency. The orange column so use nitrogen U efficiency calculated by the different methods. The method 1, and the purple column show you nitrogen U efficiency calculated by the isotope methods. You can see the isotope methods have more higher nitrogen U efficiency compared with the different methods. This is the first thing, and the second thing, you can see the urea-coated NPTVT has shows the high gas nitrogen U efficiency compared with the farmer practice and other treatment. This is also for spring dry season. This is the same result. And this table summary all indicator. You can see this is summer dry season and this spring dry season. And we have ammonia, N2O, nitrogen U efficiency, and U. And then you compare between the different nitrogen inhibitor. You can see the inhibitor SADET and NPTVT polymer have more advantage, more reduce ammonia, N2O, and increase nitrogen U efficiency and U compared with the farmer practice and other inhibitor. And this is the same for spring dry season. One again, the urea-coated NPTVT shows the high gas, the ammonia reduction, N2O reduction, and increase nitrogen U efficiency and U. So we give some comment, some advice to the farmer, and we convince them try to use urea-coated NPTVT or urea-coated polymer instead of use normal urea. Yeah, it's a suggestion to the farmer. And this is one finished experiment, and I want to jump to another study. I don't know how many times we have, Alberto and Gumelo.
Well, we could. How long do you need for showing the?
As the second one, I think just 3 or 5 minutes.
Well, we have that time. I think we can have that presentation and then 5-10 minutes questions, and we can close at 1:30.
Yeah. So if you have a question, you
Yeah,
can...
let's open a round of questions. Guillermo, if you are there, you can take the questions
Okay.
or ask yourself.
Here, make any question? No? Quynh, I have a question for you. I have several questions, but I will try to to make other people ask. First of all, I would like to congratulate you because of the impressive experiment. All of researchers that we are focused on measuring greenhouse gases. We know the heavy workload we have in the field, and particularly at your conditions in a very complex rock rice with an impressive facility, and it's very, very impressive how you manage to measure greenhouse gases, and also even applying isotopes, which is a very expensive product, you know. Yeah. So I congratulate you and all your colleagues. And I would like to ask you about the results are very interesting, about comparing several enhanced efficiency fertilizers, even including biochar. And I would like to ask you if you notice any effect of inhibitors, NBTT or DCT, on methane emission. You measure methane emissions in the experiment, and if you detected any effect inhibitors on methane, because methane, as you say, is the target greenhouse gas in rice. So this is my question.
Yeah. We for this finished ferment, actually we mainly focus in nitrogen cycle in the fertilizer system. But anyway, we also measure methane, but not high intensive, because the budget is limited. So we focus in nitrogen, but anyway we also have underlying methane, but not high intensive. I now analyze the methane result. But I don't have final methane results right now. But I will analyze methane result later, and I hope I will share with you in next step. Okay. So we will discuss how we write manuscript. I have a draft of manuscript, and I will share with you and Alberto for your contribution.
Okay, great, very great. So that would be very interesting, because possible pollution swapping from nitrogen oxide and ammonia to methane. That would be very interesting. I don't know if anybody else has a question from here or from...
Yes. Okay. Thank you, Quynh, for your presentation. Very interesting indeed. I myself am not very familiar with the rice crop farming, or neither with the specific fertilizer treatments. But as a general question, I would say this option that you mentioned as the best option, let's say, for increasing crop yield and reducing the emissions and stuff. Would you also say that is a economically effective option for the farmers? I mean, in terms of cost, is it something that the farmers could assume as compared to the other treatments? I mean,
I think it's a good question. Thank you very much. And actually, I not yet calculate the economic aspect, but I would say the cost, because the amount of nitrogen fertilizer is the same for different treatment, but the cost, I would say, is around 20% higher, more expensive than normal urea. But the yield increasing. So I think the yield increasing can compensate for the cost, the nitrogen fertilizer cost. But I not yet calculate the economic indicator.
Okay. Any other
No.
question?
No. I think that this question of the
Yeah.
the production cost of rice
Yeah.
is very relevant.
Yeah.
To understand the possibility of the adoption
Yeah.
of these new practices
Yeah.
by farmers.
Yes.
Because it is not profitable, it will be very difficult that the farmers adopt
Sure.
these new practices
Yeah.
just for the question of environment.
Yeah. Yeah.
It is particularly important, this question, of course.
Yeah.
My question is just a question of statistics.
Yeah.
What is the percentage in the rice production in Vietnam now?
Yeah.
With the traditional urea fertilization compared with the other apparently advantages alternative of the BTBT, the polymer, or the others? What is now?
Yeah. Right now, the percentage of farmer who applies the release nitrogen fertilizer, the slow release, like MBTBT or DCD, just we not have the office statistics, but in my experiment, it's just around 10 to 15%. It's still very low because some reason. Because the first thing is the price. Yeah, 20% expensive than normal urea. And the second reason I think is because the farmer behavior. When the farmer applies normal urea, the normal urea very fast to release ammonium and nitrates, and then the rice plant easy to uptake nutrition, and then the leaf, the rice leaf become green because when the farmer sees the rice leaf become yellow, and then they apply fertilizer, nitrogen fertilizer. And if you apply urea, you can very quickly to see the result, the green color, and then the farmer think the farmer thinking is good. This is the second reason why is a barrier to upscale the applying the slow release nitrogen fertilizer. So therefore, we need to have more demonstration sign to show the farmer the final effect, the final core benefit.
For the success of these new approaches, I think it's very important.
Yeah.
The extension services.
Yeah, extension
Much extension services that are in Vietnam are a very important
very
network
important.
of officers of extension to explain and to show to the farmers the advantages of these new practices.
Yeah, we have in Vietnam we have National Extension Center, and then in each province we also have the extension center in province level, and under province level we have Quynh extension center. Yeah, and whenever we have new technology, and then the extension center will carry out the demonstration trial to show about the new technology to the farmer. But problem is after the demonstration trial, and how many farmer can willing to apply new technology, and how many farmer will introduce the new technology to neighbor is very important, because the demonstration trial is limited. Even we give a lot of efforts, but the number of demonstration trial per province is always limited. So you need very good result, and then the farmer who attend in this demonstration trial will introduce, will share with the neighbor, and then the farmer after the demonstration trial, the farmer will willing to apply. I don't know how many percent they will apply. maybe they don't apply the whole packet of new technology. For example, when we introduce new technology to the farmer, we will introduce the packet. The packet meaning the different mitigation option, like not only the AW water management, but also the seed, also the fertilizer, pesticide. This is a whole packet of technology we introduce to the farmer, and then the farmer will see which one is good. Maybe they don't apply whole packet; they would apply individual mitigation. Yeah, and then when after they apply, the neighbor will come and see. Yeah, if they see the good, and then they willing to apply. This is the way the demonstration trial work in our country.
Well, thank you, Quynh.
For very nice sector, we have very big project. We call 1 million hectare of low carbon rice. 1 million hectare low carbon rice in next in next 5 years. Yeah, next 5 years.
Quynh, thank you very much for taking us to the reality of this key crop. Can you hear me?
Yeah, now we hear you.
Yeah, okay. No, I was saying that thank you to Quynh to take us to know more about this key crop and for Vietnam and for many regions in the world. So let's have 5 more minutes to what Quynh wants to show us next, and then we will close with a quick round of questions if there are any, and we close the seminar afterwards. So Quynh, go ahead.
Thank you, Alberto. For this study is also funded by IAEA, and for this study we want to test the tool developed by Edo and Alberto. They developed the tool to estimate ammonia emission, methane, and NO2 emission from pasteurized system. Yeah, so therefore we work in different countries: Vietnam, Bangladesh, different country in our group, and each country will contribute the measure data to validate the model, the tool. Yeah. So in in this study, I also conducted my PhD study. I measure greenhouse gas emission from pasteurized field in the 2 rice season and 1 maize season. For maize season, I don't measure greenhouse gas emission, but I just measure in the pasteurized season. But in the maize season, I still apply the same treatment.
Yeah, this is, let me talk a little bit more about this. As Quynh was saying, this is part of the work we have done in the agency, and we developed a carbon footprint calculator with the help of different members of the research coordinated project we were involved. And Eduardo Aguilera from, well, who was with us at CEIGRAM and now is currently at CSIC with an ERC project. He was the one coordinating the efforts to develop this co-creative work. I'm gonna paste in the chat a research paper published on this calculator focusing on Bangladesh. Okay. Thank you, Quynh. If there are more questions, is the time in the remotely? I don't see any. Guillermo, is there anyone in the room with questions?
Yeah.
Okay, I found another one.
Yeah.
Unity.
Let's go to 1 second.
This tool very important because right now a lot of rice company in Vietnam they want to claim the carbon credit,
Yeah.
and if they use this tool they can easy to calculate the greenhouse gas emission from very rice system without the method actual method. Field method, and you can save money.
Great. So my question was about ammonia. Why the emissions of ammonia are so high under your conditions? Because I was thinking before your presentation that since the rice is flooded, ammonia emissions will be very low. But your emissions are not low at all. Indeed, they are even higher than those we have here under non-flooded crops. So why do you think emissions, ammonia volatilization is so high? 30, 40 kilograms of nitrogen per hectare is quite a lot.
So I think maybe some reason. The first reason could be the temperature. And the temperature, especially for summer, right? The temperature is very high, 35 degrees. The second thing is pH value. In our soil, the pH value around 6 to 6.5.
So high, but
Not so high.
here we have 8.
And also we apply broadcast.
Broadcast application.
Broadcast application.
When the soil is not still fluid, or when the soil is fluid, you apply
flat. 2 centimeters.
2 centimeters.
3 centimeters.
And it's urea. So okay. Nice, thank you very much.
I also compare with the result from Bangladesh. The results are more or less similar. At the beginning, I also thinking the results are high, but when I compare with the Bangladesh team, the results are more or less similar.
Okay, that is very interesting. Indeed. Okay, thank you.
Well, thank you very much, Quynh, and thanks to all the attendees of this seminar. I think we can close it now. It has been so nice again to see these interesting results from a little bit unknown reality for most of us, the one in Vietnam and the one with rice fields. I pasted the 2 key papers that Quynh was mentioning. I don't know, Carlos, if you want to mention the next seminars to close.
October is full of seminars. We have one. I will open a calendar here, or maybe here. Can we see the calendar? No, not everybody. On the 17th, so next week on Friday, we have a seminar, and then on the 31st, we have another one. They are both in the viticulture sector, so you will be both very interested. And then in November, I will make some more news, but probably on the 14th and also the 21st. So for now, all that we have is October and November, but I will be informing you all soon.
Perfect. Great, thank you. Okay, thank you everyone, and have a nice weekend. And Quynh, see you next week.
Yes, Ameto. See you next week.
Bye-bye.
Bye-bye.